Hello everyone and welcome back to my YouTube channel. So today's topic for the video is important questions of pharmaceutical quality assurance, that is in your 6th semester B Pharmacy 3rd year according to the PCI syllabus. We'll begin with unit number 1. In unit number 1, basically the most important is ICH guidelines, that is International Conference on Harmonization. They basically have a guideline that is based upon these four parameters: quality, safety, efficacy, and multidisciplinary. Also, you have to remember the which Q1, Q2, Q3, Q4, Q9, Q10, Q8, which Q stands for what. Like Q2 stands for analytical validation, Q8 stands for pharmaceutical quality system, Q9 stands for risk management system, Q1 stands for stability studies. There are further classification Q1A, B, C, D, E, F based upon novel drug system, photostability, etc. So you have to remember that what is the guideline and which one it stands for. ICH, the aims of ICH is important to be remembered, and the tripartite agreement. You have to remember the three countries that are involved in the ICH guidelines and what are the roles and what are the pharmaceutical and industrial members of the committee. Besides that, you also have to remember the role of Canada. Also, you can remember the ICH different parameters for stability studies like long term, short term, intermediate. For freeze dried products, there are different conditions. In unit number 2, the most important thing that you have to remember is the plant layout and the design and the types of areas: aseptic area, cleanup area, filling area, ancillary area, production area. So you have to remember that the production area should not be close to the changing room, ancillary area, and also the washrooms, clean rooms, etc. And you have to remember that the receiving bay and the dispatch bay needs to be separate. They cannot be same, so they have to be distinct from each other. And you have to draw a diagram, a layout of the plant, so that you can explain your answer well. Next is unit number 3. In this you have GLP, that is good laboratory practices, and current good manufacturing practices, wherein you have to study about the buildings, storage, environment, cleanliness, sanitation, disinfection, the flooring, walls, doors, windows, ceilings. So whenever we go upon choosing a particular area for manufacturing, we first see the environment. What is the nearby location? What is the surrounding environment and climate? Secondly, we see what is the building, what is the structure, what are the floors, walls, doors, windows, ceilings, are there any cracks or any leakages? Next we see is the hiring of the personnel. Is the personnel trained? Is he having experience? Is he having a good amount of training, competency training? And we also do the checkup of the personnel. There are different areas like ancillary, production, manufacturing, warehousing. In warehousing also we need to keep the raw material storage different. In a separate compartment, the finished products, a separate storage area for the products that are under test, that is UT, that are going for in-process quality control test, IPQCs. And then if there are any detections detected, a separate QC and QA area should be there. QC for testing and analysis, QA for authorization, validation, and that there should be a key personnel. Key personnel is basically a person who is responsible, who is the head of the production area. That is the key personnel of the production area. Then you have the key personnel of the quality control area, and then you have the key personnel of the quality assurance area. Next, you have to remember is the quality control test for primary and secondary packaging. Primary packaging is something that is in direct contact with your product, with your formulation, but the secondary packaging is in contact with the primary packaging, that is further in contact with your original formulation. So secondary packaging may include certain rubber, certain boxes, cardboards, cartoons, etc. But the most important primary packaging is of either glass or plastic. Glass, you have to remember the four types of glasses: type 1, 2, 3, and 4. Type 1 is borosilicate, type 2 is hydraulic resistance, type 3 is also hydraulic resistance. Type 4 is non-parenteral glass. That is the type 2 is treated soda lime, and type 3 is regular soda lime. That is, you do not treat the surface with any fumes of sulfur dioxide, but in type 2 we do give a treatment with some fumes. So yeah, there are different tests like hydrolytic resistance test, water attack test, powder glass test, transparency test, particulate matter test, spectral transmission test. That needs to be reminded. For rubber also there are certain tests. For plastic, like fragmentation test is very common, so you have to remember those as well. Next, we have the good laboratory practices and good manufacturing practices that you have to study in detail. GLP mainly for non-clinical and clinical laboratories. Whenever we are dealing with animals, so we have to see to it that they undergo a proper, they are not contagious, they are not contagious diseases, they are not affected with any other harmful or toxic diseases. If they are, we have to isolate them, quarantine them, and acclimatization of those animals whenever they are bought in, and then proper use of those animals, and if there is euthanasia required, so an appropriate method of euthanasia. Unit 4 consists of SOP, that is the standard operating procedure. SOP is for everything. Even for writing a SOP, there is another SOP that is called as the master SOP. For SOP, there are SOP on cleanliness, SOP on environment, SOP of equipments, SOP of cleaning, SOP of handling the materials, everything. Then there is MPR, that is the master manufacturing record or master formula record or master production record. Then there is BPR, BPCR, and BMR, that is batch manufacturing record, batch processing record, and batch production and control record. Complaints. Whenever a product is marketed, it is very important to keep its good distribution practices, and we keep a documentation, a report, a record of where it is distributed. And if there are any complaints, we have to classify the complaints in the critical, major, and minor. If it's critical, it requires an immediate action. If it's a major complaint, it should be around 3 to 4 days. And if it's minor complaint, within 5 to 6 days we have to deal with the complaint, and it will determine whether the product is to be salvaged, whether the product is to be recalled, or the product is to be redistributed after a few modifications. So the complaint handling is also very important. There's a separate personnel to handle the complaints, and the complaints can be handled in two ways, either in a documentation or in actual testing process. So if a product is said that if it's not meeting up to the quality, it is again tested. One has to go through the records and one has to go to the documentation of the product, so that one can determine where exactly is the fault arising from. In unit number 5, we have spectrophotometry, and that also UV, that is ultraviolet spectrophotometer, in which we use UV rays, and there are different regions: fingerprint region, functional region, and IR if we go to see. But in UV, you basically determine the qualification of the UV. There are different qualifications if we go to see. Design qualification, that is called as DQ. Performance and process qualification, that is called as PQ. Installation and instrumentation qualification. There's a thin line between qualification and validation. So don't just mix them. Usually you validate equipment, or you validate, usually it is a process that is more particular, but for qualification we usually qualify the process, we qualify the design, we qualify the instrument. So these terms are not to be mixed. They need to be, one has to know the difference between the qualification and validation, and the types of validation. Either it is a regulatory validation, it's an internal validation, it's an external validation. So one has to know the difference between the different types of validation. And then there is calibration of pH meter. We use certain devices on a regular basis for calibration. So they need to be calibrated at different pHs using buffers of phosphate buffer, citrate buffer, borate buffer of different pH, 4, 6, 9, 11 also sometimes. So we use different buffers of different pH for calibration of equipments. So that is also very, very vital. And that's all for today's video. I hope you enjoyed it. And if you have any doubts, feel free to drop in the comment section, and I will be able to, I hope I'll be able to answer that. Thank you so much everyone for watching.